Good morning, children. I am here with the second part of Principles of Inheritance, and in this chapter we are going to study about incomplete dominance, then codominance, pleiotropy, polygenic inheritance, and chromosomal theory of inheritance of Sutton and Boveri. Before starting, let us recap what we have studied till now. You remember that Mendel worked on pea plant from 1856 to 1863, and he proposed three laws. First is law of dominance, second was law of segregation, and these two were derived from monohybrid, and from dihybrid cross, law of independent assortment. These were very important laws of genetics, but his work was not recognized in his lifetime. Let us see. Many scientists worked independently on crosses after Mendel. Mendel did his work in about 1865, and after that scientists like Karl Correns, he was from Germany, Hugo de Ries, a Dutch scientist, and Erik von Schirach from Austria, they worked independently on crosses like peas and of course other plants also. Let us see what they did. Hugo de Ries, he was a Dutch scientist. He worked on pea, but he worked on one more plant, that was evening primrose. And while performing his hybridization experiment on primroses, he found lot of new varieties of flowers. The varieties were quite numerous, so he came to a conclusion that these changes in the varieties are due to mutations, and later on he gave the famous theory of evolution by mutation. So Hugo de Ries performed experiment on this plant. Then Erik von Schirach, he was Austrian. His grandfather was a teacher to Mendel. He worked on crops just on the pattern of pea like Mendel, and for development he worked for the development of hybrid seeds, and he was successful in making a hybrid of wheat and rye. You might have heard of triticale. So the first experiment of this type was performed by Erik von Schirach, and of course he worked on preparing new varieties. And he also independently came to the conclusion of what Mendel had did. These three scientists, they independently rediscovered Mendel's laws, but they credited Mendel for his original work, and today we know Mendel as father of genetics. Then Karl Correns, he was a German scientist, and he performed his experiments in Germany. All the three performed their experiments in different places. So just like pea crosses, if a cross is performed between red flowered plant with a white flowered plant, what will the result according to Mendelism? This was a famous experiment of Correns, and this plant is Mirabilis jalpa. So what he did, instead of taking pea, he took this brightly colored flowered plant, and he took a red, this as red, and then he crossed these plants. Let us see what was the result. It was supposed to be, if this is dominant and this is recessive, this has supposed to be a dominant character, but it was found to be pink. After self-pollination, let us see what was the result. This was self-pollinated, and these were the pistil and the anther of the same plant. So let us see what was the result. Yes, it had red, pink and white flowers in the F2 generation. This plant is Mirabilis jalpa. One more plant can be seen for similar type of pattern, and this is similar to the experiment of Erik Correns, who was also a monk like Mendel. Let us take example of another plant, that is Antirrhinum. This is Antirrhinum. or commonly it is called as snapdragon. And Mirabilis jalpa is called 4 o'clock plant. So this is snapdragon, or children also call it the dog flower, because the flower looks like the mouth of a dog. This part children often play with it. So let us see if this is considered as capital R. So this has to be small r. And here the gamete will be capital R, and here the gamete will be small r, and fusion of these two will be capital R and small r. So this comes to be a pink flower. This is pink, and this is F1 generation with the genotype this. Now let us see what will happen in F2 generation. If this is capital R, small r, and the pistil, this is the anther of the same plant, then the gametes have to be of two types, capital R and small r. In this case also capital R and small r. This can be represented in a Punnett square. So if this is for the males and this is for the females, this is capital R here, small r here, capital R here, small r here. Let us make the total combinations. The total combinations are 1, 2, 3 and 4. Let us see what are they. This makes capital R. This makes capital R, small r. This one is capital R, small r, and this is small r. The phenotype as it is looking, this is red, pink, this is also pink, and this is white. Let us see the ratios. Come to the phenotypic ratio. What is the physical appearance? And that phenotypic ratio goes to be with red is to pink is to white. So what is the number of red plants from total combination? 1 out of 4. Pink 1 and 2, so it is 2 out of 4. White is 1, so it is 1 by 4. The ratio is 1 is to 2 is to 1. Come to the genotypic ratio. Means what are the genotypes? So the genotypic ratio has to be capital R is to homozygous R, then heterozygous R, then again recessive homozygous. This is only 1, so 1 by 4. These are 1 and 2, so it is 2 by 4, and this is a single, so it is 1 by 4. This comes to be 1 is to 2 is to 1. So here you can see that this is the phenotypic ratio and the genotypic ratio. They are same. They are equal to each other. And this codominance is exhibited in blood groups in human. How many types of blood groups are present in human beings? You have studied this in your previous classes. Yes, blood group A, then of course blood group B, blood group AB, the universal acceptor, and blood group O, the universal donor. These are the major 5 blood groups discovered by Karl Landsteiner. Now let us relate this to genetics. Now if it is blood group A, this A blood group, the red blood cells, they have a sugar of this pattern, brown, red, purple, and combination of these 5 sugars you can see is bit different from this combination. Here you find a purple sugar. The purple sugar is N-acetylglucosamine missing. So this RBC having B blood group has a slightly different sugar on its plasma membrane, and this sugar is different. Let us look at this. in form of a table. The plasma membrane of red blood cells have sugar polymer that protrude from its surface. And how do they make it different? Let us see. You can see a pattern here that these are rounded looking sugar molecules protruded from the plasma membrane, and here the ends are looking squarish. So this is the major difference between blood group A and blood group B. So blood group A has this antigen on its surface, but it has antibody for this type of sugar, and blood group B has antigen of this pattern on its plasma membrane, and it has antibody A. Now the blood group AB, it contains both the sugars, this square one also and this round end sugar also on its plasma membrane. They do not contain any antibody, but it contains antigen A and antigen B. Come to case of O. O contains antibody A, antibody B, but none of the antigens on its surface. You can see here. So this is the molecular difference between the blood group of these four types. Now what is the genetics behind this? The kind of sugar is controlled by, say, gene I. This sugar is produced by the effect of gene I, which is present on 9th chromosome of human cells. This gene I has 3 alleles. Now this is an example of multiple. How many alleles you have seen in case of the experiment of Mendel, the experiment of Mirabilis Chalpa? We have seen 2 alleles, but here you see that this blood group, this gene I of blood group has 3 alleles. Now here is again, this is something different from Mendelism. So this is one of the deviations from Mendelism. And when it has 3 alleles, we say that this is an example of multiple alleles also. What are these 3 alleles? One is IA, one is IB, and one is small I. As we have the convention of writing this is a dominant one. And now this is also dominant, and small I is a recessive allele. So the previous concept, this can produce the enzyme, this can produce the enzyme fully, normally, unmodified, but this is a modified allele which cannot produce the enzyme to produce the sugar. The blood groups you can see. Now if it is A blood group, so the genotypes are IA and IA, or it can have a recessive one, IA small I. Here it has IB, IB, here it is IB and small I. So this is a heterozygous and this is a homozygous. This is heterozygous, this is homozygous. Now what is important is blood group AB. We are studying codominance. So when we say co means together. Dominance, both are dominating. Who are dominating? Allele A and allele B. It is IA and it is IB. So this is also a dominant allele and this is also a dominant allele. So both of them are expressing themselves. So both of them are producing sugars. So one type of sugar is by A and other type is of B. So both the type of sugars can be produced. So this type of blood group makes AB. Here both are recessive, so there is no antigen. So this is blood group O. So this is making the concept of codominance, that both of them are dominant in case of blood group AB. Now let us work out a cross. What is the question? Let us see. A child has blood group O. Now when we are solving the question, read the question very carefully and write it like mathematics. So let us describe this part. A child has blood group O. So write child has blood group O. So what will be the genotype? The child has genotype small I, small I. Now the second part is the father has blood group A and mother has blood group B. Now father A and mother is B, having B blood group. Now what will be the genotypes? They can be of two types, either capital IA, or it can have IA and small I. Mother can have the genotype IB, or the other possibility IB small I. Now let us take first case. If father has case 1, if father is IA and mother is say IB, so the gametes here will be IA, IB, and the child will have blood group. Yes, what will be the blood group? It will be AB. But no, it is not AB. Let us see the second case. The second case is case 2. Now the child is having this, so let us take a common sense that this is not possible, this is not possible. Child will inherit at least one allele. So let us try this in second case. It is IA small I and mother as IB and small I. So let us make the combinations. We can have a shorter way of making these combinations if we don't want to make a Punnett square. Now carefully combine this and this allele. So it is IA, IB. Now this can combine with this. So it is IA small I. This can combine with this. We write the capital one first, IB and small I. And this can combine with this. So it is small I and small I. Now this cross does not mean that they have 4 children. This is probability. We can make prediction. Like the child, you can become a person to predict, a Jyotishi, and you can say the child can have a blood group AB, or the child can have blood group A, this is A here, or the child can have blood group B, or the child can have blood group O. So this is the possibility, 25% possibility that the child has blood group O. So if the child has blood group O, we can predict that his parents have heterozygous A and heterozygous B. This is how you are going to solve the questions which will be coming in your examinations. Make the complete cross, take a lot of space, try to do it neatly in your copies. Okay, the another concept we are going to study is pleiotropy. Pleiotropy refers to when a single gene controls many characters. You can see here, what is this? This is a chromosome. And on this chromosome, say this red colored gene is present. So it is a single gene. Single gene, a piece of DNA which should be responsible for a single character. But this is again different from Mendelism. One gene is causing a trait A, trait B, and trait C. So this phenomenon is called pleiotropy. Let us take the examples. This pleiotropy word is derived from Greek word pleion, that means more, many, and tropos means ways. So pleiotropy word is derived from Greek words. Example is a disease. Commonly we call this disease as PKU. It's not PQ, it's PKU, but you can remember from that. This is an inborn error of metabolism, and it is an autosomal recessive trait. It means it is caused when the gene become recessive. The affected individual lacks an enzyme that converts phenylalanine into tyrosine. So there is a conversion reaction in the body in which this phenylalanine amino acid is converted to tyrosine, and this requires an enzyme. But the gene is lacking for production of this enzyme. So since this enzyme is not produced, this reaction does not take place. Now then what will happen? This phenylalanine, it gets accumulated and converted into phenylpyruvic acid in other derivatives in the brain and the neurons. Due to deposition of this phenylpyruvic acid in neurons, it causes mental retardation. Then hair reduced, skin pigmentation. This is one character, this is another character, this is third character, but how many genes are present due to that? A single gene. So this is an example of pleiotropy. Let us see. This is the child suffering from phenylketonuria. So you can see that there is hair loss. This is one character. The child is looking mentally retarded. There is some pigmentation on the skin. This is also one character. Then the child may suffer from autism. He may have seizures. After incomplete dominance, codominance and pleiotropy, there is one more deviation from Mendelism, that is polygenic inheritance. You can derive the meaning from the word. Poly means many and genic refers to genes. So when many genes control a single character, then that makes polygenic inheritance. Example is skin color in human. You can see here, this is quite dark, darkest you can say. This is comparatively less, this is lesser, and these are the very fair ones. This is almost the intermediate between these two. Let us see how does the inheritance take place. The human skin pigmentation is controlled by three genes. One is A, B and C. So if it is capital A, capital A, capital A, capital A, capital A, capital B, capital B, capital C, who will have it? This one. Of course, this is the dominant character. So this is very dark, or we can say the darkest. So the genotype is having three genes here. Let us come to the lightest one. If this is the lightest one, this is very light, and the genotype is the recessive one, small a, small a, small b, small b, small c, small c. Now let us take this as a case of if there is a marriage between very dark and very light. We cannot perform a cross. We cannot perform cross like plants. We are talking of human beings. So it is matter of if there is a marriage. This can be a hypothetical case also. So if there is a marriage, if is very important, then here the gametes, and say this is the maternal parent and say this is the paternal parent, or it can be a vice versa. The gametes formed here will be capital A, capital B, capital C. Here it will be small a, small b, small c. Now when both of these combine, then the genotype is capital A, small a, capital B, small b, and capital C, small c. According to Mendelism, this could be, if it follows complete dominance, it could be dark, but practically in human population it has been observed that if the parents are like this, the color is intermediate skin, brown color. It is a mixture of these two skin types. So if these two blend, then the progeny has this intermediate color. Now here it is example of incomplete dominance. So this follows incomplete dominance. Let us see what will be the result if both the parents are intermediate type. This is intermediate type. So we have seen in the previous cross, this is capital A, small a, capital B, small b, capital C, small c. Here it is the same case, and if there is a marriage between these two, so you can see that these are the genotypes of the parents. Now the sperms formed can have any of these combinations. It is again probability. This Punnett square is showing the probability. They may have one child, they may have many children, but the possibility is that the child can have skin colors either of these. Whichever is the combination and whichever is formed as the resultant of fusion of gametes, the color of the child will depend on that. So the child can have colors ranging from very fair to very dark to intermediate, or on the darker side or on the lighter side. So these are the number of phenotypes possible. They can have either of the children. So this is example of polygenic inheritance. This does not mean that these are the number of children. This is probability, remember, because mathematics is also applied very nicely in biology, especially genetics. Such cases observed in case of wheat. You, while eating wheat, you could have observed the grains, and when your parents go to buy the wheat, they are very keen in choosing the color. So it can range from very dark to very light, the intermediate colors. This is also the resultant of polygenic inheritance. The case of genes are A, B, B, and small a, small b, small b. They control color of the grain in case of wheat. So this is an example of polygenic inheritance. Chromosomal theory of inheritance, proposed in about 1902. This was another landmark in the history of genetics. So again we are studying the history. Recall, when did Mendel propose his theories? In about 1865. But his work was not recognized till 1900. Why? Because first reason was that communication was not fast like today. We have mobiles and internet, and communication is within seconds. But in those days, the scientists did not come to know very well about Mendel's theory. Second was the reason that Mendel that the genes did not blend. So this non-blending of genes concept was not accepted in those times. Why? Because it could not explain the variations seen in the nature. The variations practically seen in the nature could not be interpreted by this non-blending of genes. So the people or the biologists of those times, they did not agree to what Mendel said. Third was that the approach of using mathematics to explain biological phenomena was totally new on those times. In those times, people knowing biology had very poor knowledge of mathematics. So this new concept was unacceptable to many of the biologists of those days. And fourth, and the most important thing that Mendel's work could not be recognized was that he could not, with the proposed hypothesis of factors, Mendel could not give a physical proof of the genes. Whatever is done in the case of science, the study of science requires evidence. That evidence can be experimental and it requires a physical proof, but Mendel could not give the evidence, so his work was not recognized. Let us see the historical course. as we have already discussed, that Mendel published in 1865 whatever was his discovery, but remained unrecognized. Why? We have seen the reasons. After 1865, it was in about 1878, a scientist, Walther Fleming, he was observing cells under the microscope. This means that till this time, cells were very well visible and very well made microscopes came into existence, and scientists were making studies on them. So Walther Fleming saw that in a nucleus, in a cell, the nucleus was dividing. He observed dividing nucleus. So this was a new phenomena in those times. Later on, it was described as what we today say is mitosis. So he had a physical proof, he had an evidence that the cells divide, and in mitosis what he saw was that the chromosomes were moving towards the pole. So this was a physical proof that chromosomes and equational division was seen by Walther Fleming. In about 1895, another scientist, Edward von Beneden, he observed cells of Ascaris, and he saw that the daughter cells received half the number of chromosomes. So in Ascaris cells he found that the number of chromosomes was half. This was later on described as meiosis. Now till this 1900, mitosis was discovered, meiosis was discovered, there was physical evidence of all these. So in about 1902, a pair of scientists, they observed that the behavior of chromosomes was parallel to the genes, and then they proposed the theory of chromosomal theory of inheritance, and the scientists were Walter Sutton and Theodore Boveri, and in 1902 they gave the physical evidence to what Mendel had discovered, and this was the basis of genetics. Let us see what was the observation of Sutton and Boveri. You can see the division. Which division is this? This is meiosis. Now, if you take this cell, how many chromosomes are present here? The number of chromosomes is 4. Let us consider that the gene for, say, yellow color. The yellow color gene, dominant gene, is present on this chromosome, and the green recessive gene is present on this. Here, this is round gene present on this chromosome, and the wrinkle gene is present on this chromosome. So this was the position of the genes. During meiosis, this pair segregated to one pole in meiosis 1, and then the centromere had split, and then this came to another pole. How? If this contains Y, this contains R, so small y is here and small r is here. Now after splitting up of these centromeres, this gets one y and small r. This gets one y and one r, and here it gets a small y and a small r. So what type of combinations are these? These are YR and YR. What about the other two combinations? Now Sutton and Boveri said that there is a possibility that alignment of chromosomes may differ. So if this is the alignment of chromosomes in first case, the alignment of chromosomes can differ in the second case. So Y has, this Y is here and small r is here, and small y is here and capital R is here. Can you see the difference? This is blue, blue, red, red, they are in the same line, and here below the blue is red and below the red is blue. So this makes the combination. In case of meiosis, this is Y, this is small r, this is small y, and this is capital R. After the splitting of centromeres, this has Y and big Y and small r, big Y and small r, small y and capital R, small y and capital R. So these were the combinations, capital Y, small r, and small y, capital R. These are the gametes which were made by Mendel according to mathematics. But this gamete formation was seen experimentally as a physical evidence by Sutton and Boveri. So what they said, that the behavior of chromosomes was parallel to the behavior of the genes, and they used chromosomes to explain this movement. You can see, or you can have a look once more here, that this gene, these are the parental types and these are the new ones. You can have a look at this video and it will show that how the chromosomes can align in.
The orientation of each pair on the spindle axis is random. Either the maternal or paternal homologue may orient toward a given pole. The number of possible chromosome orientations equals 2 raised to the power of the number of chromosome pairs. In this hypothetical cell with 3 chromosome pairs, 8 2 to the 3rd possible orientations exist. Each orientation produces gametes with different combinations of parental chromosomes.
This is comparison between behavior of chromosomes and genes. This is from NCERT and they have asked you a very interesting question, that tell which column A or B represent chromosome and which represents this gene. Let us find out. In this case, occurs in pairs, okay, they are similar. Then segregate at the time of gamete formation such that only one of each pair, this is very important, only one of each pair of what? Chromosomes. Here they mean only one of each pair. So this is the pair of chromosome, only one of each pair segregate, okay, and here segregate at gamete formation and only one of each pair. It means one out of each pair. So it is this. So this segregate and this segregate. Got it? Which one is the gene? This is the gene and this is the chromosome. So the behavior is parallel. Segregation takes place, then independent pairs segregate independently of each other. Again, they are referring to the pair of chromosomes. And here it is that one pair segregate independently of other pair. This is in case of the dihybrid, that if it is like this, so one pair segregate independent of the other pair. So the conclusion of this comparison was that what Sutton and Boveri proposed that genes are present on chromosomes. See how the knowledge developed. Mendel said factors. Sutton and Boveri said factors or the genes are present on the chromosomes. These are the genes present on chromosome. He gave the experimental proof, he gave the physical evidence of what Mendel could not prove. So this became the fact that genes are present. Let's recap the chapter, what we have studied till now. We have studied about incomplete dominance, taking the example of Mirabilis chalpa, antirrhinum, codominance with the example of blood group, pleiotropy with the example of phenylketonuria, and polygenic inheritance, we have seen the skin colors, and at last we studied chromosomal theory of inheritance of Sutton and Boveri. Now you study all these topics at home and practice maximum questions possible, and stay healthy, stay safe. Thank you very much. The world will be happy and healthy again. You'll be paid.